it's my honor to introduce our guest. I first came across him, probably in Instagram, I would say, when I first saw you, and I know you did some work with our fair, our virtual fair. Gary Heferle, I'm pronouncing that right. Okay, he is a commercial grower. You can find some of his products at local farmers market. I think you said Castle Rock Farmers Market this year, and he'll mention more about some of the other opportunities for that. But he also offers cultivation classes, which is very helpful for people who want to learn how to grow. All right, and so he's got a huge presence on social media. I was looking at his YouTube channel, like 40-some thousand subscribers at last count I saw. So it's good to see that you're getting a following. So I'm happy to see it. So we are very fortunate to have him speak for us tonight about growing gourmet mushrooms. And I'll leave it at that. So thank you, Gary. Oh, do we have another? Oh, yeah, we're good. We're good, I think. Thank you. And so those of you on Zoom, if you have questions in the chat, and we will we'll deal with those as they come. So, all right.
All right. Well, thank you. For those of you who don't know, like Greg said, I'm Gary Heferle, and I own a very small gourmet mushroom farm in Sedalia, Colorado. So it's not Salida. It's about 40 minutes south of here, between Castle Rock and Highlands Ranch. So I would like to thank all of you guys for coming out here. Usually I just talk to the airwaves on YouTube, so it's definitely a lot different talking to you guys in person. But there's a few ways that I could have gone about this talk. Normally I teach this whole course in 2 days, and it's very hands-on. But John kind of guided me to kind of dial it back into more of just a global perspective on growing mushrooms. So I'm going to cover maybe like 3 different philosophies of growing mushrooms. So one being indoor cultivation, like what I do. And then as more of like a hobbyist grower, maybe you want to just do more like gardening. That's kind of how I started. So planting mushrooms in your garden. Or there's like a third type or philosophy of cultivating mushrooms on logs. So using like plug spawn. So that's kind of the gist of this talk. So this is a picture of our new 1,500 square foot facility in Sedalia. So we started this construction back in COVID. So my wife and I, we purchased some property in Sedalia, and we were living in Denver. And I started my mushroom farm kind of as a hobby in my basement. And then slowly it evolved over the past 7 years. And I've grown and sold about 20,000 pounds of gourmet mushrooms out of my basement, which is kind of crazy to talk about. But this is me about 13 years ago. So my background is in medical technology. This was a picture of me. I was doing genetic work at Buffalo, New York, Buffalo General Hospital. I was doing some karyotyping, and that was towards the end of my college career. And back in May 2012, my wife and I, we took a road trip all over the country in my Jeep Wrangler, and we ended up staying in Glenwood Springs for a few nights, and we really fell in love with Colorado. So that October, we moved across the country, and I worked in a tissue culture lab in Centennial, and we made medical products using stem cells. So that's kind of my tissue culture experience. I did that for a few years, and then I joined this company called Native Roots when the cannabis industry became legal here in Colorado. So I took my tissue culture experience and applied it to plants, and was interested. Plant tissue culture is very similar to mushroom cultivation. So being an avid gardener, I kind of like collected a bunch of lab equipment for doing plant tissue culture. And then maybe about 10 years ago, I tried to grow mushrooms. So Um, I also completed this book, Growing Gourmet Mushrooms for Market, 2 years ago. So if you're interested, this is a very detailed outline of all of my processes, everything I'm going to talk about today, but just really dialed in. There's a bunch of links to different products that I use. And yeah, if you want to check out my website, it's freshfromthefarmfungi.com. But basically, I started off growing mushrooms in my garden. Maybe about 7 years ago, I posted an ad on Craigslist. I was growing tons and tons of lion's mane mushrooms. My wife and I, we were eating mushrooms like every day. So it was at that moment, though, someone drove their bike from Lakewood all the way to our house in Denver, and they pulled up to the front door. I had maybe like 3 pounds of lion's mane, and he looked at it. He grabbed one of them and ate it like an apple, and like I was like, no, you're supposed to cook these. But anyway, that was the moment that clicked where I'm like, there's a market for these mushrooms. People are very health conscious here. And that started Fresh from the Farm Fungi. Okay, so as a cultivator, one important aspect is what is the goal of mushroom farming? So the main goal is to produce high quality, nutritious mushrooms in a repeatable and sustainable manner. So anyone who's ever tried to grow mushrooms for the first time, it's very common to fail. My first ever experience was I bought these spores off of Amazon, or they called them spores, but in reality, it was a grain spawn. They showed up super dried out. I didn't know what I was doing, so I planted them in a pot like seeds, and then I covered it with some plastic. And maybe 3 weeks later, I finally saw some fuzz like come to the surface, and then the next morning, it was all green and it was trichoderma. So very disheartening, but at the same time, my personality was to double down. So I did a ton of research. I went down the rabbit hole. There's lots of forums and Facebook groups that I spent a lot of time to kind of procure this information. And eventually I was able to produce mushrooms. So the key to growing mushrooms is to reproduce nature in a controlled environment. So that could be a tote under your bed, or that could be a very sophisticated grow room. But all of these principles you can apply to most types of mushrooms, and you can kind of scale them to your liking. Okay, so the basic mushroom life cycle. So this is a false morel, and it's sporulating. So that's kind of the whole life cycle captured in one picture. But basically, a mushroom will start off as a single spore in nature, and it will land on a surface that has the correct moisture content. And then that spore will germinate as a haploid, or a monokaryonic cell. So it has half the genes, and it will extend hyphae, which are kind of like this long stringy substance, which once a haploid finds another compatible haploid, it will form one diploid cell, which is called mycelium. That mycelium will extend and it will cover the surface of the substrate, and then it will exude compounds that will break up the substrate so that it could absorb the nutrients, and then it will fully colonize that substrate. And in nature, oftentimes it will rain, or the temperatures will drop to condense the moisture in the air, and that will trigger pinning. So pinning is kind of the formation of that mycelium into a fruiting body. And then over the course of a day or two, that mushroom will kind of absorb water through the mycelium, and it will blow up like a balloon and produce fruiting bodies. So that's what happens in nature. It's kind of remarkable when you think about it that a little microscopic spore will land on a substance and find another mated pairing to form mushrooms. And that is kind of like the mysterious nature about fungi in general. Is I think it's a miracle that it can happen in nature, let alone in a highly sophisticated lab. So when you're farming mushrooms, the challenge is to get repeatable results in an efficient way and avoid contaminants. So when you're cultivating mushrooms indoor, there's a lot of other fungi or bacteria that also like that environment. So aseptic technique is very important, and we'll talk about that later. But I think that if you compare it to nature, when you're growing mushrooms, one of the most important things is to get really good genetics. So my passion these days has been kind of breeding mushrooms. So if anyone's interested after the talk, I brought in some cordyceps that I've been breeding over the past couple years now. But I also sell genetics on my Etsy shop, Fresh Fungi. But essentially, you want to get really purified mushroom strains in order to be able to reproduce those over time. So that comes in the form of liquid culture or petri dishes, or spores is a different option. So then you'll take those genetics and produce spawn. So it kind of can accelerate exponentially at this point, which is really good when you're growing food. But so from the spawn, you'll transfer it to a substrate, which that's just, imagine like a spongy material that the mushroom grows on that also provides nutrients. So that can be sawdust, which is the case in my farm. It can also be straw. You can use a lot of agricultural waste, which is a really powerful aspect of growing mushrooms. You can use soy hulls, alfalfa. Here in Colorado, I've grown oyster mushrooms on hemp hurd. It was almost free, so you can take a free waste product from agriculture and turn it into nutritious food. So that's the substrate aspect. A lot of other mushrooms, like portobellos and button mushrooms, they will grow on compost. So that's a different type of mushroom, but it's the same idea. Okay, so I guess the next phase after you're growing the mycelium on the substrate is to get it to pin. So like I said before, what happens in nature is that it will rain, the temperature will drop, evaporation occurs, and then that will force the mushrooms into pinning. However, when you're working inside of a building, it can get a little tricky. So for this process, I will grow the mushrooms in bags, and then I'll cut little slits in the bags, and that exposure to air will cause them to pin. Okay, and then I guess the last part is harvesting your fruiting bodies. So mushrooms grow very rapidly. They'll go from the size of a little pin, maybe like a half an inch, to the full flush like that in maybe like 72 hours. So keeping up with your harvest cycle is very important, especially when you're thinking about, how many mushrooms you want to grow in this size of space. Okay, so this next slide is going to talk about how we do this. So I kind of touched on the mushroom spawn or culture. So it's very important to get good genetics. Having, I think that there's a few really reputable companies in America. North Spore is a really good one. Field and Forest. Mycelium Emporium is very popular. I would recommend My Genetics personally because they're kind of procured in Colorado. But getting really good genetics is important because it's going to help you battle any contaminants or any fluctuations in the environment, which early on, there's a big learning curve. Okay, so then I talked about the colonization of the substrate and aseptic technique. So I'll kind of move on to this slide. So this is the types of starting cultures. So on the left there, you'll see the mycelium that is growing on a petri dish filled with agar. So this is kind of the most basic way to grow mushrooms. The petri dish acts as like a three-dimensional filter. So the benefit is that if you have mushroom mycelium growing on the surface of a petri dish plate, it will only grow in two directions. So that reveals any contaminants that might be on that dish. So it verifies that it's a clean and healthy mycelium to start with, and that will help take care of a lot of questions later on. So then I think about 10 years ago, according to the Shroomery, like one of the most famous mushroom forums, the introduction of liquid culture was made. So liquid culture is just a liquid broth. So I use 4% honey water, and it contains polysaccharides and different sugars that the mushroom needs to survive, but it's in a liquid form. And this allows for the mycelium to expand rapidly. So you can see this jar in the top right corner here. That is about a half-gallon jar of liquid culture. When I take, it could be as little as 2 cells of mycelium, so just a little scrape off the petri dish. I'll put it into that jar and then agitate it with a stir bar, and in about 4 or 5 days, I'll have enough liquid culture to generate 1,000 pounds of mushrooms. So it's a very fast and efficient way to start a mushroom farm, I would say. But there are some drawbacks. Compared to the petri dish, you can't really see if it's contaminated or not. So if you're going to start with liquid culture, just be aware you want to get it from a reliable source, and you want to kind of be cautious about it, because you don't know if it's contaminated or not until you're kind of already growing. So I would say the easiest way is the petri dish, because you can verify that it's clean. The liquid culture is very efficient. And then the third to start a mushroom farm is the spore print. So on the bottom left, spores are produced by the mushroom when they're mature in their fruiting phase. So like this picture here, you can essentially make a spore print by taking that mushroom, putting it on a clean piece of paper, or I like to use a petri dish. So I'll just take a blank petri dish, put a mushroom in there, cover it with a bowl, and let it drop its spores for about 10 or 12 hours. Then you can take the bowl off and then take the mushroom off, and you will have a spore print remaining. So that is the genetic starting point. You can kind of compare it to a seed for a plant. So there are drawbacks to starting with spores, though. So they're pretty variable. Compared to a tissue sample like a petri dish or a liquid culture, you're not going to get the same exact mushroom. But when you're breeding mushrooms like me, it's kind of exciting because there's a chance you can get a one-off like phenotype or a one-off variance from what you were growing to begin with. So there are reasons that you could use spores, but a lot of people get confused when they buy spores and then they put them into a grain spawn, because all of those spores will be competing against each other, and you won't get as good of results as if you started from an isolated culture. So the fourth type of starting culture, slant. So often if you buy your samples from a reputable like tissue bank like ATCC in Pennsylvania, they will send you a slant. Or if you buy a culture from Paul Stamets, oftentimes you can buy like a slant, which is the mother culture. So it's the original culture. It's very secure, and it hasn't experienced what is called senescence. So if you keep using the same culture over and over, it will eventually break down and kind of die out. So there's a value in a slant culture. So for my farm, I will breed mushrooms from spores, isolate a single culture, make a bunch of slants of that, and then every year I'll just take my mushroom from the mother culture slant and expand it from there. And that way I get really repeatable results over time. Okay, so after the original culture, there's a few options you can go. So this is called spawn. So spawn is basically the carrying mechanism from the pure mycelium into the bulk substrate. So on this slide, I have pictured sawdust spawn. So if you're thinking about doing a garden or just planting mushrooms in the ground, sawdust spawn is a really good way to do it because sawdust itself is kind of selective for mushrooms. So if you used grain spawn, which is in the bottom right, it's a very nutritious and it's very good for growing mushrooms indoors because it's going to give you big yields. But if you plant it outside, it's going to be competing against animals, insects, other fungus. So it's kind of important to think about what your environment is and what spawn you want to use. So the bottom left is going to be plug spawn. So I make my own plug spawn by cooking some wooden dowels from like furniture making. You soak them in some water for overnight, pour off the water, put it in a jar, cook that jar for about 20 minutes at in a pressure cooker at 15 psi, let it cool, and then you can add your mycelium to the plugs. And then a really easy way to grow mushrooms is you would take a hardwood log that has been freshly cut, and then you would drill a bunch of holes, hammer in the plug spawn, keep it moist for about 2 or 3 months until the mycelium can colonize it. And then you can bring it into a cool, damp environment that's exposed to the elements, and you'll get some mushrooms fruiting that way. So I think that that's probably the cheapest and easiest way to do it. And then grain spawn is over here. This is some shiitake on some grain spawn. So you can use a bunch of different types of grain. So I prefer oats because it's cheap and it has like an outer shell that kind of protects against contaminants, but people will often use wheat, or for the cordyceps, I grow these on rice. So the idea of a grain spawn is that it has a very high moisture content. So you can really blow up those grains like a balloon. And if you think about mushroom farming compared to other agriculture, irrigation happens up front. So you can't really add water to mushrooms after they start to fruit. So the value behind grain spawn is that it holds all this water up front while the mushroom's still developing, compared to sawdust, which that will probably hold about 45 to 55% relative moisture compared to grains, which they can hold up to 65% moisture. So that little bit of difference will mean, that much more yield. Okay, so now that we talked about the starting culture, intermediate culture, there's a process from going from a sterile media to a less sterile bulk substrate, and that is called inoculation. So this is kind of like the hands-on part of my class. You can't really learn it just by reading about it and thinking about it. You kind of have to do it hands-on. So that's where the process of aseptic technique comes in. But basically, once you have your spawn, you have to transfer it to a growth media, and you want to be able to do that without introducing contaminants from the air, like yeast or mold, or just like bacteria from your breath. So in this picture to the left here, that's my wife, and we're taking a liquid culture that we brewed inside of a bottle, and then she's taking a sterile needle, and you can see there's this injection port on top of the jar. So it's essentially like an IV that would allow for you to transfer the mycelium into that sterile vessel without introducing it to the air. Another tool that a lot of mushroom farms use is a flow hood. So that's a laminar flow hood that she's working in front of. It's taking the air, it's running it through a HEPA filter, and it's filtering out any potential contaminants. So these are all things to consider when you're trying to grow mushrooms. You want to have it as sterile as possible so that you'll achieve the next level of growth there. Okay, so yeah, this slide kind of talks about the aseptic technique. So the goal is using sterile process to maintain the isolation of mushroom mycelium and prevent contamination. So you can use a still air box, or nowadays they make these inflatable glove boxes on Amazon for like $100, and it basically just protects the mushroom from different, wind currents or like your breath, like I said. Okay, so this is especially important during the early process, like if you're starting from spores or from a petri dish, and then as the mushroom develops, it kind of gains immunity and it gains strength where it can defend itself against contaminants. Okay, so this um, kind kind of stepping back from the actual process of mushrooms is the space that you want to be able to grow. So I would say all of the techniques that you can grow mushrooms require some kind of a lab space or clean space. So this can be like a glove box, like I said, but this right here is my lab up top. I have a flow hood that I do all of my culture work in, and then I have another incubator that after I do my petri dish work, it maintains the sterility. And then uh, stainless steel tables that are easy to clean. This lower picture, I'm showing you the injection ports that make it easy for you to take a needle and just kind of expand your mushroom cultures. So the less you have to handle your mushroom spawn, the better, the more success you're going to have. Okay, so then after you inoculate your spawn or your bulk substrate, you're going to have an incubation space. So if I was doing this as a hobby still, I would probably consider a closet a good incubation space. It's going to be dark. It's going to be relatively stable environment, and it's going to be free from, other contaminants getting in there. And then one of the most important aspects is that it has to be easily observable. So mushrooms grow very rapidly. Things can get out of hand very quickly. So if you don't observe your mushrooms every day, then problems can happen. So if you ever do see, a green spot or like a oily substance on the bottom of your spawn, it's probably contamination. So you should take that, um, isolate it, watch it, and then if it's, continues to get worse, then you can compost it, but you want to remove it from your incubation space. So having protection from contaminants and also protection from direct light. So mushrooms, they typically grow in the undergrowth. They don't like direct UV lights. So these are all considerations when you're thinking about the space to grow. Okay, so now from this point on, the mushrooms start as a culture, then they develop into spawn, and then you have this bulk substrate, and it can kind of divert in different directions at this point. So you can either grow in outdoor garden beds, like that top right picture, which is I'm growing King Stropharia. So they tend to grow well in just a garden environment, outdoor on logs, like this picture in the top left here, or indoor in a controlled environment. So essentially, up until this point, it's the same. But then once you get to the point of actually fruiting the mushrooms, that's kind of where you can differentiate the technique. So I really love growing King Stropharia in garden beds. I've grown it with squash, corn, um, tomatoes. And typically what I do is right now I have about 30 pounds of spawn that is ready to go. And once the weather kind of stabilizes and it's time to plant, I would say like your early season vegetables, like corn is a good one. I like to till the earth and then put my spawn down and make like a lasagna layer. And in this picture here, this is hemp hurd that I got from a friend for free, but you can use leaves, you can use grass clippings, you can use straw, or just any kind of like a high cellulose material. And then you'll just want to plant the spawn in layers so that over the course of the spring, the mycelium will thicken. And then as the plants grow, they'll provide shade. And then probably around, late June, early July, you can get a flush. It is kind of variable depending on the weather. But yeah, usually I'll see 1 or 2 flushes throughout the summer. And in my opinion, the King Stropharia tastes very similar to a porcini, but you don't have to go out and hunt them. And there's usually less bugs because you can just pick them right when they're ready. But I recommend, starting with King Stropharia. Okay, so then I also talked about planting in logs. So it's pretty simple, but basically you would take a fresh cut log, preferably hardwood. So in Colorado, I've had success with cottonwoods, and you want to you want them freshly cut and not diseased. So there's a company called Chip Drop. If you guys aren't familiar, um, make sure you're prepared. But basically, you can sign up for this service where local arborists in your area will put you on like a map, and when they do tree trimmings, or if they have to like clear a bunch of trees, it's kind of indiscriminate, but they'll come to your house and drop off a big load of wood chips, or sometimes they have really nice logs. So that's what I've done in the past, where I've gotten really nice cottonwood logs. But you can also use the wood chips to grow King Stropharia. But you would take these logs, wait maybe a week until the sap inside the wood will kind of dry out, and then take a drill, and every 4 inches put a hole in the log, and then put a colonized plug spawn in that log. And then some people say you should seal it with wax. I think that it's unnecessary, but it will help the log kind of stay moist. And then what I like to do is I'll stack the logs for about 3 or 4 months really tightly together and put a tarp over it, because in Colorado it's so dry that mycelium could dry out. But after about 2 or 3 months, after looking and seeing the mycelium, so you can kind of see the mycelium coming out of the end of that log on the left, then it will be time to flush. So you can either just drench it with a hose for 10 minutes, and then wait, and then maybe the next day do it again, or you can dunk it in like a bin with water. But the idea is to get that log really saturated. And then as that log will dry out, you should get pins. And then in about 2 or 3 weeks, if you're growing shiitake like this, you should get a nice flush. And then the benefit of logs is that they're very like slow burning, so to say. So you can get years of mushrooms out of a log compared to sawdust, which breaks down really rapidly. Okay, so then the third aspect, which I kind of talked about a lot, is growing indoor. So these are Piapino mushrooms, and I think they're one of the easiest mushrooms to grow. You can grow them inside of a grow bag. There's a company in Texas called Unicorn Bags, which they make a biodegradable plastic bag, which is pretty awesome. And it has a filter patch. So you would take that bag, add your sawdust, which I use a 50-50 blend of hardwood and soy hull pellets. And then I add water and cook it in a barrel, like a steamer, for about 24 hours, let it cool, add my spawn, and seal it. And then you don't have to do anything else but just watch it grow, because the bag is kind of designed for a little bit of airflow. And at least the strain that I bred, it will pin on its own just right on your countertop. And then once the mushrooms get to a certain size, you can just cut the bag open and harvest them. And if you preserve the integrity of the bag, you can kind of put the flaps back over and get about 2 or 3 more flushes like that. So I have some videos on YouTube about growing Pinon mushrooms. I think they're the easiest to start with. Also oyster mushrooms. But yeah, so then this slide kind of shows different parameters. So if you do check out my Etsy shop, Fresh Fungi, all my strains have these growing parameters, which kind of go through the expectations for timelines, and I think temperature. Yep. And then I do have like a whole PDF file. If anyone wants to email me with questions about these, I could send you all this info. But personally, I grow about 16 different varieties, and then I rotate them throughout the year, just depending on the weather. So, okay. Yeah. So normally, like I said before, I teach this class in like 2 days and it's very hands-on, but I'm kind of here to answer any questions. So I don't know if that was kind of a good overview for you guys, but yeah, I can answer questions if anyone has more specific questions to growing mushrooms. Otherwise, I did have like another part of fungal ecology to talk about. But yeah, if anyone has any questions about growing mushrooms. Yep. Yes. So yeah, I really like them. There are some drawbacks though. So the technology behind the biodegradable bags is that they add an additive to the plastic, which makes them vulnerable to oxygen. So as soon as you get a shipment of those bags, there's kind of like a shelf life where it's about 6 or 8 months before you have to use them or they degrade. So I'll have to order them more often, which kind of increases the cost a little bit. But the idea is that the plastics will break down, and I have a small compost pile where there's it's slowly like degrading. One question I always have is like, what happens when they get really small? Does it become microplastics and stuff? So I haven't like dove deep into that research, but there's a company called Pegasus Bags, which is one of the employees at Unicorn created this side company just based on that. And I also talked, or in my class, I touch on this plastic eating fungi. So it's a P microspora, which Pegasus Bags gifted me one time. And they isolated this mushroom in the Amazon, and it breaks down polypropylene plastic or polyethylene plastic. It's one of the two, and that is like, half of the plastic that they use. So there are some flaws behind it, but to me, it's a lot better than, you know, the traditional plastics that they're using. It's like a step towards the right direction. Yep. Yep. Go ahead.
This fungi will break down to like biodegradable plastic or any kind of
Let me look. There's a slide I have. Okay, I was wrong. It breaks down polyurethane plastics. And the research that they did was in like a bioreactor. So it wasn't exposed to any oxygen. I think it was anaerobic digestion. But this is, the forefront of these plastic eating mushrooms. I think that they also discovered another species like maybe 2 years ago, and that has more promise as far as like being exposed to oxygen. But basically, they starved this mushroom until its only option was to eat plastic, and then it broke it down. So I don't know how you feel about that, but I feel like it's like a step towards the right direction, but there's a lot of research still being done. Yep.
repeat the question.
Okay. Yeah. Yep. All right. So anyone else? Yep. Go ahead.
A similar idea. Do you know of any mushrooms that are being developed to like target like deadfall pines and deadfall like aspens?
Okay, so the question was, are there any mushrooms that are being targeted for deadfall pines and deadfall aspens? So I think personally, Raguso annulata So Stropharia, Ragusa, and Annulata, or the garden giant, that has been able to break down any of those wood chip drops that I've gotten, which is, 40% pine. There's even like walnut in there sometimes. So I think that King Stropharia is like this hybrid between like a secondary decomposer and a primary decomposer. It's very adaptable. I think that that would be a good candidate for it. I also think that oyster mushrooms, there was a company here, Grow House, and they claim to use softwood for their brown oyster, or Pleurotus austriatus, maybe. I don't know. One of their oyster mushrooms, they only use softwood pellets. So I think that the challenge for like fallen wood would be that there's like a differential, or there's like a differential in the moisture content of like a log that's laying on the ground. So I think that breaking those down a little further, or burying them so that that moisture content would be more stable throughout the whole log, would help it break down faster. So I think that's the challenge when you have like a bunch of debris piled up, is that if there's too much like surface area to the air, the mushrooms will only grow inside that log, if that makes sense. Yep. So, yep, go ahead.
Made petri dishes or slants at home. How long can you keep them for?
Yeah, so the question was, how long can you keep petri dish or slants when you make them at home? So I think that there's a few factors in the shelf life. So the first one is, are they sterile? So what I do as a normal procedure is, when I pour my plates or make my slants, I'll leave them at room temperature for at least 72 hours. That way, if there is any contaminants while you're making them, they would reveal themselves. So that instantly eliminates, however many you contaminated. So I think that starting off, 50% is really good success. But nowadays, I maybe get 0.1% contamination. So that would eliminate right off the bat. Then the next factor is, if you use a sealed container like a slant, you can potentially keep those for a decade if they're not contaminated and they don't dry out. Now, with petri dishes, there's vented petri dishes, so those will naturally kind of evaporate, especially with the surface area being so large. But you can use glass petri dishes, and as long as they're kind of sealed and you have some weight on it, those can also last for a very long time. Now, there's some media types like rose bangle agar that is sensitive to light, or if you have different antibiotics in your agar, it could degrade over time. But if you're just using malt extract or potato dextrose agar, it could potentially last for a very long time. Yep. Okay. Yep.
Follow to that. Would you have to store them in the fridge, or is it going to be stored
Yep.
room temperature or freezer, or what?
Okay, so the question is if you should store them in the fridge or room temperature or a freezer. So I would say not a freezer, because when they thaw, they'll kind of get misshapen. A refrigerator is best practice. So any lab like that's like CLIA certified, or any regulated lab, the reason is that the nutrients won't degrade, or like I said, if there's antibiotics and stuff, it will maintain the viability. And if you store it in a fridge, my recommendation is to store it upside down, so it's agar side up. And the reason is that when it condenses, all the water will be on the lid of the petri dish. So if you have condensation, you could just take the dish and pour that off without getting the agar contaminated. But that being said, if you keep it at room temperature, just make sure that it's like not like warm in the room. Like if you have like a 90-degree room, it's going to cause the agar to evaporate. But if it's like 60 degrees in your room, that plate will slowly get thinner, maybe over the course of a year, 2 years. So the temperature is a factor because of evaporation. So the water from the agar will condense against the surface of the plate, and then it will shrink that down till it's just a little film. But that could take years or decades, just depending on the temperature. Yep. Yep, go ahead. Yep. So the question was, how did I get into this or mushroom farming, and how long have I done it? So if you missed the very beginning, it kind of started as a hobby. So I started off by trying to grow mushrooms in the garden. I remember a phone call I had with my mom one time. I'm like, yeah, I'm gonna grow mushrooms in the garden. She's like, what? Like, you can't do that. And like, being like, my personality is like, I'm very anti-authoritative, I guess. So yeah, so I was like, okay, I'm gonna try growing mushrooms in the garden, and then I completely failed. And then I started researching, and then the first successful mushroom that I grew was actually from a grow kit that someone left on their porch on Craigslist. So my wife was at work one day. She's like, hey, I found this like post on Craigslist. I'm like, I drove there instantly, grabbed this like, it was like a dilapidated oyster block, and then I put it in this little green, it was like a tent that I don't even remember, but it was like this little 4-foot tent, and I was like spritzing it with water, and eventually I just like added a humidifier, and I got this mushroom to flush. And then maybe like 2 months later, I went back to New York, and my dad, one of his friends at work, her husband had a mushroom farm in Niagara Falls. So his wife would always like bring in mushrooms just to give to people. And it's like a very Italian culture where I'm from in Buffalo, New York. So, we like mushroom lasagna and mushroom pizza, and it's just part of the cuisine. So I always kind of was interested. I just didn't know the cultivation aspect. And then luckily, I was there on Thanksgiving, and he was like shutting down for the season. So he offered me to do a tour, and I was like, heck yeah, I'm gonna go. So I went and visited. I talked his ear off for like 2 hours, and he was kind enough to just show me his whole process. And then I ended up leaving with a grocery bag of mushrooms. And I was driving back to Colorado. So we like put ice in a cooler, and I kept these mushrooms viable. And then as soon as I got home, I cloned all of them. And that really took it to the next level, because I had like 8 different strains, and I was constantly like transferring them. And at that time, I was working with a lab for the CDPHE. So I was lucky that I collected like all this lab equipment. They had like an old incubator and an old autoclave, like this little 2-foot autoclave. And I was just, I'm like, you're gonna throw that out. So I started just collecting stuff. And then that was probably 7 years ago now. So then I saved up a bunch of money, and I ended up quitting that job after I started. It was June in 2018. So maybe in February, I started posting mushrooms on Craigslist. And then in April, someone actually bought them. And then in June, this lady, Christina Welch, was starting a co-op grocery store. So she saw my ad on Craigslist and was like, do you want to sell your mushrooms here? I'm like, yeah. So then that allowed me to grow as many as I could. And then slowly, I kind of was selling out of mushrooms there, and then I could produce more than I was selling. And then the following summer, I joined the Cherry Creek Farmers Market. So that was kind of like another like breakthrough, because I couldn't plateau there. So that was maybe 6 years ago. And then 2 years ago, we built our new building. And then now I've kind of maximized that, but I'm shifting more towards breeding mushrooms. And I was telling Greg earlier, one of my goals is to like almost automate breeding. So that's kind of like my scientific background as a med tech, meeting like my passion in mycology. So that's kind of the future where I'm going from the beginning. Yep.
Zoom questions. What do you do with all the spent blocks after fruiting?
Yep. So the question was, what do I do with all the spent blocks after fruiting? So when I first started, I bought 2 tumbler composters, and for a while I was composting my mushrooms, and then I would bag it up and sell the compost. And a few years ago, the the gardening guild at one of the farmers markets approached me, and they would come and take truckloads of compost. But ever since we moved to our new facility, I have this enormous compost pile, and I'm contemplating what to do with it. But right now it attracts like 50 turkeys because there's like, yeah, like the grubs that grow in there are crazy, and like it's just interesting to watch. But I feel like it attracts lots of wildlife right now, and my hope is to, make raised beds and kind of garden out, and then once I have a certain, I'll probably sell it to the public again, or if you're interested, just email. But yeah, people can come and take plenty of it, but it's a good byproduct for having a mushroom farm. Yep. Oh, go ahead.
If you're interested in growing a diverse variety, would you only have one fruit chamber? What species of varieties would you recommend?
Yep. So the question was, if you only have one fruiting chamber, what mushrooms are compatible with each other? So if I go back to this chart, the text is super little, but I tried to like pair up mushrooms together, but I can just tell you what I do. Um, so in the early season, like right now, I like to grow king oyster, blue oyster, brown oyster, lion's mane. Those are like my 4 starters. Those all like colder temperatures. And then as I drift into like summertime, I'll switch it up and I'll do like shiitake, because those take longer too. So unless I started them in like December, they're not going to be ready. And then I also do like chestnut mushrooms, piapino, golden oyster, pink oyster. Those are very heat tolerant. And then as I switch towards like the really warm parts of summer, lion's mane always thrives. So that's kind of like a all season one. And then also beach mushrooms, they can handle the warmer temps. And then I feel like I'm missing a lot. But so one thing that you have to think about is if you have one fruiting chamber, there's microclimates within there. So I I even started just with one tent, and I would put my lion's mane all the way at the top, and then something that could handle a lot more moisture, like a shiitake, you could put that right at the bottom next to the humidifier. So even if you just have one fruiting chamber, try to divide it into different microclimates and just play around. But also genetics plays a big factor. Like some oyster mushrooms are very tolerant to CO2. So that's another factor that I didn't even talk about here. But if you keep your CO2 below 1,000 ppms, most of the mushrooms that I sell will grow fine together. It just really depends on your airflow, your humidity, and like the proximity of the mushrooms. Like if you squish too many close together, they might have problems. Yep.
This might be more of a business type question, but how do you manage succession planning to keep up with your market?
Yep. So the question is, how do I manage succession planning to keep up with my market? So that is a very um organizational. I don't okay. So at first, I would say my bottleneck was like I had space problems. So just one little area of space, created my bottleneck, which that kind of limited my time. Now that I have so much space, I think labor is kind of my bottleneck. So what I do is I divide up my week into different portions of the grow. So Monday, I'll do my bulk sterilization. And then while that's cooling on Tuesday, I do my grain prep. And then both of those will kind of cool by Wednesday. So I'll do my inoculations on Wednesday. And then Thursday and Friday, I kind of package my mushrooms for the farmer's market. And then during that whole entire time frame, I'm constantly picking mushrooms and putting them in the cold storage. So it's really just staying consistent and having like a routine. It's very difficult to step away from the mushroom farm. So I have a couple friends that will babysit it for a couple days, but after that, it gets out of hand. So that's why I chose to do a seasonal mushroom farm as well. Yeah, that's Nolan. Yeah, but yeah, I think that it just depends on your schedule. Hello. All right, any more questions?
Another question was asking about your book.
Okay. Oh, yeah, so the question is yeah, about the book. So yep, so I wrote the book with the intent that it was like kind of during COVID, so there was a lot of like online learning, and it's has a lot of links to videos and links to products that I use, but it's essentially a start to finish guide on how to grow mushrooms for markets, and kind of like at the scale that I was at. So about 100 pounds a week, which that's about 60 hours a week of labor. So kind of balance that out. But yeah. And then like I said before, I do teach hands-on 2-day cultivation courses. We have a couple spots left for our course in April. And then I'm teaching an outdoor gardening course in Arvada in May. And then I think nothing until potentially in the fall. I was trying to do some kind of DNA class, but it's still pretty hazy at this point. But yeah, so the book is basically a guideline on how to get started. And then if you want to learn more hands-on, we do teach classes at our farm. And then we also do farm tours too. So that can all be found on our website. But yeah, all right. Up, go ahead. Yep, so the question is, do I mention breeding or did I? Yeah, so yep, so I kind of touched on it a little bit, but basically breeding is there's a few different methodologies. So the classical method of breeding mushrooms is to take spores and streak them out on a petri dish and then select the strongest culture that kind of emerges. One thing that I like to do is to isolate single spores. So you can do that by diluting spores, or you can do that manually under the microscope. And then the advantage of isolating a single spore is that there's a chance that you can kind of crop out a rare type of genetics that would otherwise be kind of outcompeted if you did it traditionally on a petri dish. So sometimes when you streak out spores, it could take 6 or 8 days for one spore to germinate. Where if you isolate a single spore and put it on a dish, and you could still capture that rare genetic. The problem is that it's very labor intensive and it's very material intensive. So in order to get, this is my 30th rendition of cordyceps, and it took about 2 years and maybe like 400 petri dishes just to get this little amount of success. where if you're operating like a commercial farm, it's probably not the best allocation of resources. So kind of having a balance between that is my philosophy, but also like I was saying earlier, my vision is to automate that process. So there's a lot of like automated pipetters nowadays you can buy fairly cheap, like $2,000-3,000, and you can kind of hone in that aspect for isolating spores, and then maybe make it more efficient. But yeah, so in nature it just happens like randomly, and then up until recently, it traditionally was just you take some spores up and just rub it on a petri dish, and then the strongest one survives, and then you select that, and then you select that. But I think that you can almost like create a prism of genetics by selecting single spores and then combining them. So I'm working on that, and I'm kind of focusing on my next book, which will be more about breeding and selecting genetics and that kind of thing. Yep. 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 So the question was, what am I looking to find for when I'm breeding? So as a mushroom farm, I'm looking for yields, fast growth, resistance to environmental changes, taste, like texture. Some mushrooms look really good, but then when you cook them down, they're like super hard. So that's like an undesirable trait. Um, just like appeal. So at the farmer's market, everyone loves pink oysters because they just stand out and they're like, what is that? So creating like unique phenotypes like that. Yep. All right. Yep, go ahead.
You have, thanks for the very educational presentation.
Yep.
You have not mentioned reishi.
Yep.
Have you ever tried grow them, and any challenges, difficulties, and why not?
Yep. So the question is, why didn't I mention reishi mushrooms? So I do grow them. I think they're very interesting. I just didn't really think about it, but so the challenges of growing reishi mushrooms are that they're very slow. So usually I'll start them maybe like now, and then they'll be harvested in like late August, September. But they are a very unique mushroom because you can grow them in a high CO2 environment, and they'll kind of grow like antlers. And then if you introduce them to more oxygen, they'll kind of conk out and give you like the classical fan appearance. So there's a really talented grower called the Fungi Florist. If you look him up on Instagram, he creates art out of reishi mushrooms. I don't know how he does it, but he makes like zigzag ones, and very, very interesting grower. I recommend, just checking his grows out. And the cool thing about reishi mushrooms is you can kind of preserve them just by like letting them dry out. It's almost like a form of art, like I said. And then what I'll do is I'll take like a branch off and then grind it down to make a tea out of it. And it's, there's a lot of medicinal properties. I just think that it's kind of more of a niche mushroom. So I have brought it to the farmer's market before, and people will buy it, but it's very rare. So, yep. Yep, go ahead.
Like any nutritional differences, like mushrooms grow on the inside, wild. Do you like ever mess around with different nutrient additives besides like soy?
Yep. So the question is, are there different nutrients? Is there variation of nutrients between an outdoor mushroom and an indoor mushroom? I would say yes. I haven't done like the official testing, but my instincts were when I first started growing mushrooms, I would work with this company called CS Woods, and they were like a specialty wood maker. So they would import all these like trees from Sedalia. And I noticed like batch to batch when I was growing on some exotic hardwood compared to just like a cottonwood that I got from up north in Broomfield. There is a difference in flavor and texture. So I personally think there's probably a difference in nutrition, but I never did the testing. But I think that as my farm kind of developed, it was so difficult to like isolate different types of sawdust and stuff. So then I just eventually converted over to pellets just because it was easy to reproduce over time. But maybe someday I would revisit that and do like micro batches of different wood types. But I think that there's definitely a difference. I kind of like glossed over it early on, and then it's kind of hard to maintain that viability at the scale that I'm at. Yep. All right. Is that about time? All right. Well, thank you all very much. I enjoyed answering your questions and talking mushrooms. So thanks for coming out here. And if you see me down in Castle Rock, we do the farmers market every Saturday. We also sell to a crop share program in Arvada. You can find all the information on our website. And yep, I'm sure I'll be at some meetings here soon too. So thank you very much.